I am Kamal, the director. I would like to acknowledge and thank our administrators who are with us today. Scott Furlong and Kristin Croyle. They understood the role Rice Creek plays in education, academics, in outreach and research. And we are really grateful for the support of the field station. I would also like to thank Rice Creek Associate, all of them, and especially Rice Creek Associate Board of Director, who are with us today. So I'm going to name them the present Board of Director in no order, just to thank them. Laurel and Dan Artz, Mike Flanagan and Mike Holly, Sherry, Maury, and Pat Jones, not sure they're here, Paul Little, our student rep, Brooke Goodman, Andrew McElwain, who is actually taking the job of the small grant presentation, and also Andrew sometimes come up with those really funny jokes. Thank you, Andrew. Who else? And then Kristin Haynes. I want to thank her and her magic fingers also, and Wendy Fraggle, who is probably watching now. Did I miss anybody? I think there's about 11 members. They're very helpful for us. They leave everything and they come to the field station to do the work of the field station. So we thank them for that. So for example, they were meeting here yesterday and they were here today, and also they're meeting on Wednesday. So they are really, really dedicated group of people. So we are grateful for them. So for today, we're going to hear actually a word from our provost, Scott Furlong, followed by our dean, Kristin Croyle. By the way, our boss said we have to use the microphone.
The boss says. Good afternoon, all, and welcome both to SUNY Oswego and to the field station. What a blessing we have here at SUNY Oswego to really be able to take advantage of this wonderful site, and a double blessing because we can really take advantage even in November this year of this site without getting snowed on or anything. When I arrived here a little over 5 years ago and was able to see this space for the first time, I was really blown away by the opportunities that it presented, and really by the role that Kamal and the faculty and now Kristin have been able to continue to grow it in a programmatic way and to work with our students, because what we're really all about at SUNY Oswego is providing great hands-on opportunities for our students to interact outside of the classroom in whatever way they think is appropriate, and whether it's undergraduate research, internships, service learning, whatever the case might be, and to be able to come out here and really get some really dramatic and significant hands-on experience is really again something not every institution can claim. So I want to thank all of you for coming today to see some of the fruits of those efforts, and also thank the board for their continued support for the Rice Creek Foundation, and the funds that are coming in to help our students do the work that they're doing. So thank you very much.
Okay, well, he covered all the bases, right? So I will just add, if I haven't had the pleasure of meeting you, I'm Kristin Croyle. I'm the Dean of the College of Liberal Arts and Sciences. I will just add that, you know that Rice Creek has both a very focused and at the same time a very broad mission, right? And that each of us touches that mission in different ways, the way that we use the facility, the science that we participate in, the community activities that happen here, the beautiful trails and grounds. I just want to mention, are you aware that they're doing over 300 events a year? I think I know you know the exact number, and that is an underestimate. 300 is an underestimate. So thank you for all your wonderful support. I'm looking forward to the presentations, and thank you to the staff and to Rice Creek Associates for continuing to push forward the mission of this beautiful facility and location.
Hi, I am Laurel Artz, and I am the president of Rice Creek Associates. I just want to thank you all for coming and showing your support for our researchers and all the wonderful work they've done. Our job as Rice Creek Associates is to assist the Rice Creek Field Station and the things they do, and essentially with the research. And we have granted, let's see, since 1996, we have had over 100 projects totaling over $135,000. So we feel pretty good about that. And I hope that you will enjoy the presentations today. I don't want to take any thunder away from Kayla, but on your way out, make sure you stop in the herb garden and take a really good look from both sides. You'll see what I mean when she talks about it soon. But go take the time to walk over and look from both sides. That was a project we've been trying to do since before COVID, and we finally accomplished it. She accomplished it for us. The way you can help is become a member of RCA. Our dues are very reasonable. There's pamphlets on every table. There's other pamphlets describing things we do over here. Please consider becoming a member and supporting what we do and supporting the research of our students and faculty and the greater community here. So thank you.
Alrighty, I'm Andrew McElwain. I'm the Small Grants Committee Chair, and I will be introducing each speaker. So Kayla, would you like to come up? Your presentation is loaded onto the computer.
Yeah,
And so we just need to share the screen here. And then talking to the microphone if you can.
oh, hello.
And there's a laser pointer here if you need that.
Hello, everyone. My name is Kayla Kitchener, and I'm a senior BFA student here at Oswego. And today I will be talking about the sculpture that is getting installed into the herb garden. This is my first ever public commission piece. So I'm super excited to take you through my process behind the design of the piece, as well as everything I did to get it here today. So this is the entire digital design that I proposed to Rice Creek back in May. You can see that there is a front panel, a back panel. This is the edge, which goes around here, and then it leads to the bottom. So when I was brainstorming the design for this, I wanted to focus on the activity that goes on inside of the herb garden. And after looking at some of the resources that Rice Creek provided to me for the proposal, I found that the garden is extremely important to insect life here. And then I decided to highlight as well the importance of the native species in upstate New York. The first thing that I had to do was include the Oswego tea flower for obvious reasons. And then I also wanted to highlight the honeybees and some recognizable insects like the monarch butterfly and their respective caterpillar and cocoons. Including the life cycle of the monarch was important to me to again point out the importance of the plants and to the insects of the garden, sorry. But as you may have noticed, if you've seen it at all, there are a few changes to the original digital design. So after talking about my design with some of the Rice Creek faculty, I decided to add a few other insects. The orange mint moth, sorry, I forgot I had to hold that. And the hermit sphinx moth that is now on the front of the new design. So my experience with Rice Creek has mostly been walking the trails that they have here. So I knew that I wanted to include that within my design because I'm sure a lot of other people have that same experience. And I also just thought it was a really cool organic thing to include within the design, as well as like the color coded, I don't know, it fit really well. And to continue the theme of native species, I incorporated some common animals that you may see while walking the trail. You can probably recognize most of these. See a cardinal, a painted turtle, a blue spotted salamander, and a cotton-tailed rabbit. And finally, the edge and bottom include yet another native plant species, the Virginia creeper, which winds up and around and down to the bottom of the design. I also included this interactive aspect of the hands. I thought it would be a cool photo op and just a way to get people sort of interacting with the piece. Now I'll be moving on to the actual process of how I made this thing. So the first element that I started with was laying the tiles. As you can see, I was able to print out a scaled up version of my original design, which helped me keep the actual piece as similar as possible to the original. I started with the color, and then I filled in the rest with some cream and white tiles. This was definitely the easiest part of this project, but it was also very tedious and time consuming. This is what is underneath all of that mosaic. It's a steel structure that I made with lots and lots of measuring, welding, and grinding. This was my first time working with metal in this way, so it was one of the more challenging parts of the process. There was a bit of a learning curve, but after more than a month in the metal shop, I am pretty confident with my welding at this point. And you can also see in this photo that, or you may be able to see, it's all gray, but this is a bit of a different gray, and it actually has Rust-Oleum on it to give the steel more longevity. And the durability of this piece is super important thanks to the wonderful winters that we have here. So that's just another aspect. After I finished the tiling and the inner structure, it was time to put it all together. So to give the tile something to attach to, I used cement board. Sorry. These are typical for tile installation, and a lot of the times you can find these in like showers or bathrooms in general or kitchens. And after bolting them on, I filled the seams with mortar in order to ensure that waterproof surface. Then it was finally time to put the tiles on. The procedure for tiling is all basically the same. First, you set the mortar, and that gives the tiles something to adhere to. You let that dry, and then you fill the rest of the empty spaces with grout, and that holds the tiles on there. What you see out there and what you see right here is the finished product basically. So we actually brought it over last night and it was pretty rainy. So we haven't fully installed it. So all that's left now is to bolt it there. So it's again, very durable, and seal the grout. Despite all the hiccups that I've come across while working on this project, I'm so happy that we got it here safely for this event. And I'm really proud of the final result and super grateful to have had this opportunity. I have lots and lots of thanks to give to everybody who helped me. I really couldn't have done it without them. First, Ben Ettner, my faculty sponsor, he teaches sculpture here at Oswego. And he got me this opportunity in the first place and also gave me plenty of insight on to how to best build this structure. Rich Bush, who works in Shinneman in the metals shop, he kindly let me work there for basically the entire semester, and he taught me a lot about the ins and outs of a good weld. Steve Ginsberg, he works in the Tyler Hall wood shop and helped me with the assembly of this thing. He was there day in and day out the past couple weeks, and I really couldn't thank him enough for all of the time that he spent with me in order to get this done. And finally, Rob Raby, who owns Raby's Ace Hardware here in Oswego. He offered his box truck in order for us to get it here yesterday. I could go on and on about everyone who helped me during this process, but that would probably take up the entire time. So I'm gonna end here. Thank you. I don't know if we're doing questions now or later, but.
Question. You can say question in the chat if you want to address that.
Oh, can I do it right here? Oh. Thank you, Wendy. I would also like to thank Wendy. She has been really helpful keeping me in contact with everyone here, as well as setting up everything and making sure that it goes well. So thank you, Wendy.
Great question. I just have never been able to work with something at that scale. How much does it weigh?
My guess is 300 to 400 pounds. We actually, yesterday when we were getting it off the truck, we had to carry it, and we basically carried it all the way there. That was with like 7 people. So if you can figure that out, that's about how many people it took.
It was really there.
Yeah, he was here.
So I just would like to acknowledge, I think actually since my last 8 years, this was the hardest project. And I think Kayla worked really hard on it, and she's able to overcome a lot of problems to bring it to this actually what we see today. So thank you very much. I mean, she did not disappoint herself or our advisors or our academic associate. Thank you.
Thank you. Sorry, I'm making a lot of noise about it.
No problem. Very well done.
I took this with me.
Not sure if this is being shared because
You could share the screen.
yeah, where's the
It maximized the little window that Ellie did.
Yeah.
Just that one.
Yeah, maximize that.
this one?
Yeah.
Right there.
Okay. Okay.
There you go.
All right.
Hello, everyone. I'm Geetha-Loganathan from Biological Sciences. So I'm here to discuss about my long-term ongoing project, which is on characterization of fungal pathogens that I isolated from snapping turtle eggs. This is actually a branched out project. I'm a developmental biologist who research on signaling processes involved in embryogenesis. So when I joined SUNY Oswego, the project I have initiated was to identify the genetic basis for having turtles developing shelled bodies. So as a part of this project, I have collected snapping and painted turtle eggs from the region around this building. And this was my first field work I've ever done. And then I was excited to get those eggs, bringing them back into my lab, incubating them and getting embryos, as you can see in this picture, collecting them at different stages in development and studying the genes that are involved in shell's body development. So while we were doing that, in 2014 summer, we found that one clutch of snapping turtle eggs, they got colonization from some microbes, as you could see from the images in here in the bottom panel. So the microbial contamination pretty much altered the shape of the eggs. It also made the yolk solidified, and the young guy developing inside got infected with the fungus or maybe some of the microbes in association. As a result, we pretty much lost the whole clutch. And when I say clutch, to give you an idea, snapping turtle egg clutch is around like 40 to 50 eggs. So if the infection is found in the clutch, the whole lot is lost pretty much. So we won't be having hatchlings developing from them normally. So when it occurred for the first time, we sort of ignored. Maybe I thought we try to have the box not maintained properly while incubation process, but we had a reoccurrence of at least like 2 or 3 times in the same season. So that kept us worried. And here you could see a normal development, and you at the embryonic stage I will be collecting is around like 4 days. And that's a happy embryo, and here is a happy hatchling closer to hatch, pretty much highly vascularized. But if you see the infected egg in here, you could see the dead embryo inside, and this one is pretty much of no use. So this posed a problem not only for the project, but also in general, we were thinking like it's just threatening the snapping turtle population, because we know that fungal infections are the major cause for declining population in different animal species. So it's been found worldwide. And also sea turtle population do have fungal infections identified, and pretty much caused the similar effects we are seeing with snapping turtles. But it's not been reported in snapping or painted turtle because it's very commonly seen in the area. And so we were not sure what's happening. So the project I'm talking today, we initiated in 2014, and the questions we try to address are whether we'll be able to isolate and characterize the type of microbial colonization we are seeing in snapping turtles. And if so, where the infection is coming from, what's the source of the infection. And to add on, I said like we do collect painted turtle eggs. We never saw painted turtle eggs to be affected by such type of microbial infection. So it's not seen until this season. We never had any issues with painted turtles. So we were wondering, like, is there any structural characterization that we will see in snapping turtle eggshells that possibly led or maybe facilitated the penetration of microbes from the egg surface. And all these together, we want to see whether this is a threat, whether this is a potential sign of threat for the population to decline later on. So we tried to first address the very first question we had, trying to isolate the microbial contamination we found on the eggs. So we had our ex-colleague, Sophia, who was a mycologist. So she helped me to isolate and to culture and to get the cultures established. And you could see that we have the fungal colonies developed within like 36 to 48 hours after getting the swabs from inside and outside of the eggs. And from there on, we went to do the characterization morphologically and to see what type of fungus is that. And in the picture, we are seeing bright field and SEM, scanning electron microscopic analysis of the fungal cultures isolated from the plate. And the techniques that are specific for fungal characterization has been standardized in my lab. So it took a while for us to get those cultures, especially for the scanning electron microscopes, micrographs. So the features we observed with these cultures, first they had branched hyphae. And within the hyphae, we had segmentation, which are often multinucleated. And we also saw conidia that are sort of comma shaped. And we also had spores that are similar to rice. And all these characteristic features, when you compare that with a fungal atlas, we could pretty much narrow this down to some species within the Acromycete fungal family, fungal class. So then we want to consolidate or confirm what type of species is that. So we went and did the molecular characterization. So for this, we did the usual way of PCR analysis using fungal specific primers, isolating the amplicon. As you could see from the gel in here, each of this isolate is individual clutch that have been infected. So we got these sequence, and the sequence analysis proved that all these belong to two species of Fusarium fungus, Fusarium solani and Fusarium keratoplasticum. And when we're observing the fungal culture plates, we also saw like tiny little bacterial cells, and it's very common for fungal hyphae to carry bacterial contamination of that. So we tried to get a pure clone of those colonies, and I have done that with the help of Pete Newell in the department. And he helped me to get those colonies grown, and then we went on, did the sequencing just like for the fungus, but this time we used bacterial specific primers. And then we identified two genus of bacterial species like Bacillus and two species of Pseudomonas groups. So from these results, we were able to determine what type of infection is seen in the snapping turtle eggs. And then followed by this, the next question is where this came from. So obviously the first question we would like to rule out is whether it came from the laboratory condition we are using, or if it has been transmitted from the adults, either from the moms during the exolate, or it's been transmitted from the males to females sexually, and then subsequently into the eggs while laying. And the third possibility is, of course, the nesting site. So soil can have those fungal species as well, right? So the very first one can be ruled out immediately because the same laboratory conditions have been used not only for snapping turtle eggs, but I also use the same conditions for incubating painted turtle eggs, Amazon river turtle eggs I get from Syracuse Zoo, and it's also commonly used for chicken eggs I use in lab and for my course. So, and also even between the snapping turtle clutches, it's not seen in all the clutches. Only some clutches are being infected. So we could pretty much rule out the first possibility. So we went and moved to answer our second question. For this, we collected swabs from cloacal mucus of adult snapping turtles. I should accept that I haven't done anything with this part of the project. I'm not that courageous to hold a snapping turtle. So it was all my students who have collected those swab samples, but I'm good enough to hold painted turtle. So we tried to get the swab from the adults of both the turtle species just to compare and see what they have, whether it's been transmitted from the adults. And I should mention a big thanks to Dr. Friesen Rosenbaum, because I haven't done anything of this sort. I'm a lab mice, and then he helped me to put the traps on, and he guided me through the whole process of collecting those samples. And we went and analyzed those swab samples. We established cultures using them, and we did the morphological and molecular characterization, just like what we have done for the egg samples. And then this time we had used confocal and different stains to do our morphological analysis, and we could clearly see all the characteristics pretty similar to the Fusarium species. And we have also confirmed it by gene sequencing data that these infections are, of course, from the Fusarium isolates from the SAP samples. And how many of the adults do have it? We collected in total 8 samples from 8 adults, 2 females and 1 male, and all the 3 had the fungal colonization showing Fusarium infection. And none of the painted turtles did have this infection. So that clearly shows that it might be transmitted from the adults. And to follow up on that, we went and collected soil samples from the nesting sites. So the hypothesis is, even if it's been transmitted by the female while laying, usually the females do urinate to make the soil moist when they're laying the eggs to dig the nest. So it can be easily got into the soil and from there, onto the surface of the eggs, or it can be present in the soil itself. So we did the soil sampling, and then again, we established the cultures, used those, and then it took really long time for us to get this part done, because to get a pure cultures or individual clones to be cultured from soil, it's really difficult. As you know, like we have tons of microbes present in the soil. So we standardized those techniques, and it took almost like 4 to 6 months for us to finish this part. And then now we know what to do. And then we did the morphological characterization. And of course, all those samples we collected from almost like 20 different nesting sites. Everything did show fungal species that has characteristics of Fusarium or Acromycete family. We are right now proceeding to do the molecular characterization for those cultures. And taken together, to answer our question 2, yes, the infection has been transmitted only to the snapping turtles from the adults and or from the soil nesting sites. And we don't see that in painted turtles. I would like to underline that. So our third question pretty much based on that rationale, why it's not seen in painted turtles, even though it's collected from the same region, just around the building here. So the answer or the hypothesis might be the structure of eggshell of snapping turtle might facilitate the penetration of microbial infection. So when we are looking at any reptilian eggshell, we should see the shell units, just like what we are seeing in the SEM pictures here. And we should also see the spores in between them to allow gas exchange and also to get water in during development to support embryo through hatching stage. So those pores can be easily measured. So we were wondering if the pore size of snapping turtle is big enough for the fungal hyphae or the spores to get inside. So we tried to get the SEM images of eggshells of both painted and snapping turtles. And then we did quantification of multiple specimens for, multiple biological replicates for both. And then we also compared between eggshells at different developmental stages, because it's known from previous studies that the eggshells do expand because they get moisture when the development happens. So after a while, you'll see those pores normally bigger than when they're late. So we try to get them at different developmental stages and we compare. The results we got here, number 1, in snapping turtles, when we have those spores compared between uninfected and infected eggs, the infected eggs had like way bigger pore size, pretty much like almost 40 micrometer, which could fit like 20 different fungal hyphae at a time. So it's really big. And then even the normal egg, which are not uninfected, the size of the pores are way bigger than the fungal hyphae or even the fungal spores. So the fungal spores are maximum of 2.5 micrometer in width. So that means it can get into it. Whereas for uninfected eggs, it's almost like 21 micrometers. So it's like really big compared to the fungal hyphae or the fungal hyphae that is germinating from the spores or even the spores itself. And how does painted turtle do with this? Painted turtle pores are the maximum of 5 micrometer. So that clearly tells us, yes, even if the nesting sites do have these fungus present, painted turtle eggs don't carry them because their pore size are much smaller than the hyphae that's germinating from the spores. So yes, the morphological structure of the eggshell of snapping turtle facilitates the penetration of fungal infection into it. So the last question, is this a potential threat? And from the data collected so far in 5 years since I joined SUNY Oswego, I will say the answer is yes, because totally we have found 6 of 11 clutches collected contain this infection. That means they are all lost. And this counts for almost like 56% of total eggs collected so far. And the occurrence of infection happens within like a week after the eggs were collected. That means within a week, the embryo is going to die. So the maximum I could get is like early stage embryos from these eggs, but it's really hard because you could see yolk is really solidified in these cases. So taking everything together, conclusions we could derive, snapping turtle population, at least in Rice Creek, have been identified with infection, microbial infections. And reason for that is mainly the Fusarium species from fungal fungal family, and then 2 species of bacteria, Bacillus and Pseudomonas infecting them. And this caused major issue for the hatching success in development of those embryos. And the source of infection is identified to be from the adults, turtles, and or soil in the nesting site. And the structure of the eggshell of snapping turtle itself induces or facilitates the process of infection. And this is the first report on such fungal infection in snapping turtles. And the next thing to address is whether it is just in Rice Creek or it's also widespread in Oswego and in New York. So that's our next step to do. So yes, we are seeing a potential threat for the snapping turtle population, causing mass mortality of the clutches. We are going to lose like 40 to 50 eggs in one clutch, just like that. So for this project, I would like to thank all my students who have worked in this. Everyone except Mohamed have graduated, so I need to present today. And I thank my collaborators. Special thanks to Rice Creek faculties for helping me to identifying the nest, and they call me immediately whenever they see a turtle nesting. So thanks to everyone. And of course, the funding sources, Rice Creek Associate Grant and Challenge Grant helped me a lot to progress through this project for at least like 7 years now. So we are close to finishing up this part of the project. So as I said, like we are going to check whether this is just in Rice Creek or it is widespread. So we are going to keep working on that. I tried to brief everything here. I didn't went into the methodologies or the details of the project. So I'd be happy to take any questions you have. Thank you. Yes. So we did the collection from 2014 to 2018 during May to June every year. And then I was not able to do the collection for 2 years because of family emergency and also because of COVID. And then we started collecting last year. And then, so in total, like almost 5 years. So we did like 11 clutches. Out of that, 6 were found to be infected. And when we are collecting the eggs, we are not collecting all the 40 from each clutch. We will be collecting maybe 10 from each clutch just to leave the natural reproduction in this population. So we'll just maybe collecting like 10 to 12 eggs per clutch and seeing infection in all of them. So that means that the whole clutch is lost. Okay, thank you. Oh, yeah, sorry, Kristin.
Hi, thanks for the presentation. My question is this Fusarium fungus, is that native to North America, or do you know?
We see it everywhere worldwide. Yeah, it's pretty much seen in water bodies, in there's associated. Even here, we see it in amphibians. I'm not sure about the bats. Maria is not here today, but I know it's available. It's seen in amphibians. So it can be from the food it's intaking. Thank you.
All right, next we have Samuel Kerr from SUNY ESF coming to talk to us about his project. Let's see here.
Is this a pointer and a clicker? Like, can I switch slides?
I'm trying to hide the floating media controls here, but. All right, yeah, just talking to have also.
Alrighty. Not working. Can you hear me? Is it good? Okay, I can't tell. All right. So, hello everyone. My name is Sam Krebs. I'm a master's student, second year master's student at SUNY ESF in Syracuse. If you're not familiar with that school, don't worry, you're not the only one. It's a very small school. It stands for Environmental Science and Forestry. Okay, so it's right next to SU, right next to the Carrier Dome. And so today I'm going to be talking about my master's project. And this is, anyone know what kind of bird this is? You got any birders here? Anyone brave? Northern flicker, excellent. Yes, so one of our native woodpecker species. So this is northern flicker on some Staghorn Sumac, which is going to be a plant I'm talking about today. And so my project, this is the running name. It might change, it might not. It's winter foraging ecology of birds. They used to be and mammals. We're just focusing on birds now on Staghorn Sumac in the central New York and Finger Lakes regions. Okay, so I'm just starting my field season. As you can see, it's winter foraging. So I'm going to be sampling from now. I actually get to go out in the field and I'll be back here on Monday. And I will be sampling all the way until March. So a lot of what I'm going to be talking about today is background info, stuff I saw last year during our trial field season. I don't have any finished results. So you're not going to get much of that, but I'll tell you what we're doing and what we've been seeing so far, all right? Okay, so today is going to be all about birds and food, all right? So hopefully you ate before coming here today. If not, grab a snack because you're going to get hungry, right? Two of my favorite foods up here, pizza and tacos. We're not going to be talking about this kind of food though, right? That's what people eat. Today we're going to be talking about what birds eat. So basic ecology, surprise, surprise, food's really important, right? I think this is probably a no-brainer for most people. You've probably seen food webs way back in like 9th grade bio or 10th grade bio. You know, this is something that's been in textbooks since the 40s and 50s. It's a pretty basic concept. So food relationships, right, are very important in ecological communities. Probably all of you could have told me that, right? So evolution of flowering plants or angiosperms in particular is responsible for today's diversity of all sorts of animals we have here in New York, right? So plants are the base of terrestrial food webs. Marine and aquatic food webs a little different. Plants play a part, but not nearly as important. And so, you know, everything, all animals pretty much rely on plants at some point. Even predators, you know, there are indirect relationships that ultimately they're eating something that's eating plants, right? So we're going to be talking a little bit about plant and bird diversity. So plant diversity, so the number of plant species, their abundance relative to others, directly increases animal diversity. Again, this is something that took years and years for scientists to kind of show through studies, but your farmer next door could have told you that, right? The more plants you have, the more birds you're going to have. So in particular, woody plant, so our angiosperms are trees, right? And bird diversity are strongly linked. So we're going to be focusing on berry-producing plants today. So berry-like fleshy fruits are a major source of food across all sorts of diverse habitats across all continents except for Antarctica, right? And so richness, or just the number of fruit species, affects the richness of frugivorous, or birds that eat fruit, bird species. All right, so now we're going to switch gears really quick because this is also really important to my project. We're going to be talking about bird vision, and this is really cool. So bird vision, there are two types of bird vision. There are those that can see ultraviolet light and those that cannot. Most birds can see ultraviolet light. Surprise, we don't. If you do, that's really cool. I can't. So human eyes only have 3 different types of color receptor or cone cells. bird eyes have 4 types of cone cells. And so this shift between just seeing violet light and ultraviolet light happened at least 14 separate times throughout the evolution of birds. So this isn't something that just happened once and kind of stuck around and was just like, oh, it was a lucky adaptation. This is something that happened 14 separate times throughout the lineages of birds as they evolved, and it is clearly beneficial. So, okay, so most birds, I said not all, are tetrachromatic, meaning they can have 4 different types of cone cells. So the typical bird eye can see all the way from 300 nanometers to about 700 nanometers. UV, the near visible UV range is about 320 to 400 nanometers. We cannot see that. And so as you can see in this diagram, that gray curve, that first curve on the left, that is the UV sensitive cone. And so the peak reflectance that these birds can see is different for every species. Most songbirds, and that's what I study, peak around 370 nanometers. So this is really neat. All right, so on the left, that's what you and I see. That's what humans see. No UV light. And so on the right, that's what scientists think birds are able to see. So this bird, there are a lot of birds that males and females look identical to us, right? So species like the American crow or the blue jay or the black-capped chickadee, we think males and females look the same, right? And then other birds like mallards, the boy's got the nice green, bright feathers on the head, and the female is camouflaged so she can hide on the nest and survive, right? Well, scientists are figuring out birds might actually be able to tell the difference between males and females in those species like the American crow, where we just see black feathers. And so they're a lot more colorful and a lot cooler than what we can see. And so what is this UV vision used for? So courtship, like I was just telling you about, the males, this is a barn owl. And so you can see that, it's a very drab, color on the plumage. And when scientists shine black lights and they're trying to work in the field to identify the age of birds and the sex of the bird, they can actually use a black light to see these UV reflecting patterns in the wings, and they can actually ID the sex and the age of the bird. So really cool. Nest incubation. So up there in the top right corner, what we see, the nest looks camouflaged, the eggs are camouflaged. This is shorebird eggs. Mammal predators like coyotes, foxes, raccoons, they really have to rely on smell. They can't just see them. Something like killdeer, a local species we have, they go and they fly away from their nests when there's a predator nearby to distract them. And then they can easily find where that nest was, even though it's camouflaged to us, because those eggs are actually reflecting UV light. And so another thing, prey identification. So this is an American kestrel with a vole in it. So, you'll see red-tailed hawks, kestrels, all sorts of birds of prey sitting up on telephone wires, right? And, we think, wow, that's gotta be really hard to see like those small mammals they're catching. And what scientists are finding out is as these small mammals move around in the fields, they actually leave behind UV reflecting urine and scat trails and odor trails. And so the kestrel is actually able to sit up there and follow the trail like a little map and then swoop down right where that mammal is. So really, really neat stuff. And then what I'm focusing on is eating fruit, right? So this is a black-capped chickadee, which I saw right outside behind you. So they're around, and this is Staghorn sumac, and that's the plant I'm focusing on. So there's this idea in scientific literature of fruit color being an honest signal when it comes to birds eating fruit. So an honest signal is one that basically lets an animal know this is beneficial, and it actually is beneficial. There are other signals that the bird is attracted to, but might not necessarily benefit the bird. So fruit color has been shown to maximize the intake of necessary nutrients and energy during peak important seasons like during migration and during winter when they're burning a lot of calories and desperately are in need of nutrients. So this bird on the right, that's the European blackcap. It is well known that that species uses UV reflectance to pick fruits that are essentially more nutritious. And then fish crows, another native species we have here, they are able to use UV reflectants to select blueberries. They're just easier to detect. So they save time in foraging and they're able to get more nutrients. And so that bottom image is just showing, you know, that's what we would see on the left. No UV reflectants. The berries are camouflaged in with that leaf. On the right, they show up better. So focusing in on my plant, Staghorn Sumac. So it is an early successional shrub, and you'll see these, they're technically called panicles, but I just call them seed heads, right? These seed cones. And so they're just clusters of berries. And you'll probably see this species all along old agricultural fields, along the sides of highways. It does really well where it's not densely wooded, you know, where there's a lot of disturbance. You know, stuff like tilling for agriculture, that kind of thing. Really, as long as the farmer doesn't mow right over it, it'll dominate in those areas. And so it contains many beneficial compounds. I list a bunch here. You know, it has a lot of nutrients for the birds. Pretty much most fruit producing species with seeds do, right? A lot of protein, a lot of fats. But it's specifically what we're going to be concerned about is these things called polyphenolic compounds. Okay, so we'll get more to that in a second. And what's interesting about Staghorn Sumac is that this isn't a trait that's unique just to sumac. This happens in other plant species, but it's relatively rare that the seeds actually benefit. So they are more likely to germinate after they've been passed through the digestive tract of a bird. So after they go through the crop and after they go through the gizzard and they get excreted, right, then they are more likely to germinate. So that's pretty cool. So that's going to kind of get into the why we think there might have been coevolution going on between birds and sumac. So wildlife use of sumac, this stuff is great. All right. So if my project is a bust and what I'm trying to show doesn't happen, you still gotta love sumac, okay? So that's what I'm gonna try to tell you all right? So all sorts of stuff, love sumac, all right? So deer, if you're a deer hunter, or if you just love deer, I just love deer, this stuff's candy to them. They will just chew it right to the ground, right? And then, you know, young shoots come up, they love the young shoots. They'll, big bucks will rub against the older stuff, but they really just like to eat the young growth, right? And then also Eastern cottontail, right? So a species I was told we have plenty right here in Rice Creek, right? So they will browse on the young shoots, like that picture there. You'll see, you know, rabbit browse, and you know, when it's tough in winter, they'll resort to this stuff for nutrients. And then, you know, there's another northern cardinal, another species that's common here. I didn't even know that when I put this on there. Look at that, it worked out. So, you know, all sorts of birds eat sumac. A lot of native and our European honeybee, but a lot of native honeybees and carpenter bees will pollinate the sumac flowers, and there are some that will actually, the wood is relatively soft, so they will actually drill little holes and nest in the sumac stems, which is really cool as well. And you know, it's not just songbirds that eat this stuff. Game species, grouse and wild turkey. I've actually seen turkeys up in the sumac, just pecking along the berries. You know, it's relatively small, shrubby tree species. They don't really roost in it, but they will eat it, and they seem to really like it. All right, now we're going back to chemistry. Okay, so polyphenolic compounds, I'm not going to get super into what these are, but I want you all to know that they are ubiquitously distributed, meaning they are across all plant species. Every single plant has some sort of polyphenolic compound in it. And so they're bioactive plant chemicals, and they are a defense mechanism to herbivory. So they basically deter things from eating them. All right. So that's what they do. And they also play a role in inhibiting seed germination. So the seed will not germinate when there are high concentrations of polyphenolics. When these things break down, now the seed germinates. This is where we kind of get into the idea where, well, if the seed benefits by passing through the bird, and these things are, you know, inhibitors of digestion. So I mentioned here gallic acid, caffeic acid. Those are the two polyphenolics I'm going to be looking at for their concentrations. They inhibit starch digestion. So birds aren't going to want to eat these things when they're high in phenolics. And they also inhibit seed germination. So the idea of coevolution happening between this plant and songbird species The idea is birds benefit when these things are low, and the seed also benefits when these things are low. And so the plant would want, quote unquote, want, right? Plants don't make decisions, but through evolution, it would evolve to have the birds consume these things when these phenolics are low, right? And so the phenolic ring absorbs UV light. So this is where we circle back to bird vision. So UV light, when these phenolics are here, when they are in the berries are not reflecting because that ring absorbs, right? So reflectance is going to be low when these phenolics are in high concentration. When they break down, the berries would reflect more, right? So birds would be able to see it. That's the idea. And then they decline throughout winter because these things are sensitive to changes in temperature. So the observed pattern we've roughly seen throughout the past couple of years, no scientific study, just what we're seeing in our backyards, is that these, and some studies have already been done, unpublished data, but some general studies have been done through honor students at Oswego to just look at some of these concentrations of phenolics. They haven't put the pieces together with with the berry reflectance and bird foraging, but we know that these phenolics decrease throughout winter. And then we also know that birds tend to avoid eating sumac until later in the winter. We'll see stuff out at that picture of that northern flicker I showed you at the beginning. I took that 2 weeks ago. So it's on it, it's eating it. But in general, most birds hold off. These things don't get decimated until February, March, usually. So the question is, why are birds holding off on eating this, right? And so the idea is they can either A, see UV reflectance and they're waiting, quote unquote, waiting, right, for that decrease. Or, winter's a tough time, right? Snow covers a lot. As winter goes on, other food sources are decreasing. It's very likely that other food sources are just running out and that they just resort to these berries later in winter because they honestly don't have another choice and they're starving, right? So there is other options as to what's happening. But our hypothesis is that these birds are using UV cues to detect whether these phenolics are declining, right? The bird's not going to be like, oh, when it's reflecting, that means caffeinic acid and gallic acid are low. It doesn't know that. It just knows from trial and error, okay, now my tummy doesn't hurt when I eat this thing, right? So that's the hypothesis. And if this is happening, the visual cue of the reflectance would be an honest signal. So if I pair up data showing UV reflectance, polyphenol concentrations, the reflectance going up, concentrations going down, and bird foraging increasing, that would mean this is an honest signal, and they know that the food is safe to consume. So I have 5 different sampling sites. I have them across central New York and the Finger Lakes. 3 of them are public, 2 of them are private. 1 is right here in Rice Creek. And so here's a map of the sites, right? So I have them in 4 different counties. I have 1 near Baldwinsville in northern Onondaga County. I have 1 in the town of Jordan. I have 2 really in the Montezuma Wetlands Complex. I have 1 in the northern Montezuma Wildlife Management Area by the DEC. I have 1 at the Seneca Meadows Wetlands Preserve, and then I have 1 here. And so what am I measuring when I come to these stands, right? So a few different things. So polyphenol concentrations, I don't measure in the field, but I grab samples for. Same with UV reflectance, I don't measure those in the field, but I grab samples for. And foraging activity, that's what I'm really measuring when I'm at the site. I'm sitting there, I'm watching birds eat it for half an hour at each site. I'm counting the number of species on it, the species that are actually eating it versus just sitting on it, right? Birds like to just perch and chill. So it's not always just eating it. And how do we quantify foraging level? I have these, berry round ranks. This is just something I drew quickly on my iPad to kind of give you the idea. So that top left one, that'd be a rank of 0. It hasn't been eaten, right? And then, it goes all the way down to 100% foraging where all the seeds are gone on that panicle, and that, that would get a browse rank of 5. And then I'm also counting berries on the ground, right? Because birds knock a bunch of these berries. If you have watched birds eat at your bird feeder, you will know they knock seeds everywhere, right? So they are sloppy eaters. So, they'll knock berries off when they come on these things and when they are eating. And yeah, and then just, I already mentioned birds, number of birds observed, the species. All right, so data collection logistics. So I'm collecting data, I started at Halloween, and I will be collecting data all the way until March. So I'm getting two different groups. I have a control group, an experimental group. The control group, I'll show a picture in a second, but that's going to be berries that the birds cannot forage on. I have exclosures that I'm making. That's something new this year that we kind of figured out last year during our trial run, that would be something really beneficial to have. So I have these exclosures. I'm going to sample berries from those once a month, and then I have experimental berries, which are just the panicles, the seeds that are just exposed to birds. And I'm going to be, coming to Oswego and digesting these samples in the lab. And I have a master's student here and an undergrad that's doing this for her honors project, and I help them in the lab. We get the phenolic concentration using HPLC or high performance liquid chromatography, and I'm also bringing the berries back to campus in Syracuse at ESF. I'm running them under spectrometer to get UV reflectance. And so this is some pictures of the exclosures that I'm making. So it's really crude. I just went to the hardware store and just bought some fencing mesh, some zip ties, some rope, right? Just put those together. I have a latch so I can open it. So the idea is these berries are exposed to the exact same temperature and weather conditions as the berries that are exposed to birds to be foraged on, right? And so I just hang them in a spot really close, and they'll have the same weather, the same temperature, everything except for the birds actually eating them. And then this is some pictures from the lab. So, some of our technology is still old and crude, right? Mortar and pestle, we got to grind down these berries. But then we have some really high-tech stuff too we do that I don't completely understand. That's the chemist stuff, right? So I have a great group at Oswego, and I'm so grateful for them. So this is Joanna, the undergrad. She was in the lab for 7 hours on Halloween Eve, right? So we were in there from 5 to midnight, and I just kept telling her, go home, go home. She's like, no, I'm gonna help. I'm like, thank you. All right. So it was great because I wanted to know what I was doing. All right. So and then this is a picture taken just this past March. So this is, what the panicles look like at the end of the season. Not necessarily everywhere. We did see variation across the 5 sites I had, but this picture was taken in that site that we have in Jordan, and the birds there just absolutely decimated 95% of all the panicles by the end of the winter. So they do eat them, right? Whether they eat them because of what I think they do, that's to be determined. But birds like sumac, and that's what I want all of you to take away for that. So a special thanks to the Friends of Montezuma. So the Friends of Montezuma Wetlands Complex, they're a great group. They're part of the Audubon. They help fund me. They also have some great events like the Montezuma Buck Race. If you're at all a birder, even an amateur like me, they're a great group to meet. They're a great group to work with. Seneca Meadows, so they have that awesome wetlands preserve that I highly recommend going for a hike if you're in the area. A lot of really cool stuff there. They help, I have a site there, and they also give me funding. The Eaton Birding Society, that's who I'm pictured with here. I just gave a presentation to them on Monday. They're a great group that's super interested in, anything bird research related. And then, of course, you all, the Rice Creek Associates, right? That's why I'm here today. Thank you. And also the DEC for letting me work on their property as well. So at this point, I will take any questions. Okay, so thank you. Fire away, don't be shy. And I will take questions later as well. Yes.
In the enclosed one, your control one that you put in the wire mesh, is that still attached? Is that a live?
That's a good question. So that's actually something we were debating, right? Because they're not attached anymore, because in order to, it would have been really difficult to make. I have to have 18 panicles in those exclosures. It would have been really difficult to make a wire mesh exclosure to just put around 18 separate panicles on every tree. And a lot of these trees, I have this big pruner, extendable pruner I need to get like 20 feet up in the air with. So the idea is plants, for the most part, pretty much all plants here, right? Because, we have upstate New York winters. So, down south in the tropics, this is different. But here in the Northeast, plants really stop sending stuff up in the fall, right? Nutrients, the flow of stuff. That's why leaves come off. That's even conifers where their needles don't come off. They generally stop sending stuff up. So the idea is they actually start sending stuff down during the fall. And so that's why if you're trying to control an invasive plant species, you actually want to pesticide it in the fall and cut it because it'll bring that pesticide or herbicide, I should say, that herbicide down into the roots, right? So it's taking all its nutrients, all the stuff, and pumping it down into the roots for over winter. So our hope is, and our idea is, we were able to cut these things off a couple of weeks ago because in theory, the plant shouldn't be changing the contents of the berry anymore. They're done flowering, the berries are mature, they're just sitting on there, and you'll actually be able to tell the difference of, these panicles don't just fall off like the next year. Old panicles, old berries will stay on, but you'll be able to tell like what was last year's berries versus this year's berries because the new ones are much brighter, they're red color, the old ones get really nasty, brown looking, they have like insect pests usually in them. So you'll be able to tell the new growth from the old growth, and our hopes are it's done sending everything, we cut them off, we put them in these exposures, it should be the same stuff happening on the live ones and the ones that are cut. Good question though, yes, excellent, yeah.
Are birds the only seed dispersal method for the plant?
That's a good question. I'm sure, the occasional gray or red squirrel or chipmunk or whatever's going up in the tree, I'm sure it'll help disperse the seeds by just knocking them around and stuff. As far as I know, mammals don't eat the seeds. If they do, let me know if you see one. I certainly haven't. The mammals really like the bark, but as far as I know, birds are pretty much the only ones that are, really eating them and distributing them far away. Any other questions? Good, both good questions. Thank you.
Just a comment. All, as a rule, all of the red Sumac are edible, including this one. So it made good tea. The other thing, dormancy, that's normal in plants, especially in our area. If you're a plant and producing seeds in summer or fall, we don't want your seed to germinate because they will die during the winter. So that's a normal thing. And also seeds passing through the birds to germinate, that's also very common, especially in the parasitic plants like the mistletoes. More or less all of the mistletoes go through this process through the birds.
Great, thank you. Anything else? All right, I think I'm all set. If you have any other questions later, let me know. Thank you all.
All right, next we have my student Jordan Meeker, who is presenting research results from her honors thesis that she did this summer.
Hi, my name is Jordan, and my Rice Creek project was the prevalence and intensity of Helminth parasites in wild and domestic canids from fecal samples at Rice Creek Field Station. Which one?
Switch on the left side. I don't know.
I could just use the
I
computer.
guess just press the buttons and then.
The mouse.
I broke it, I'm sorry.
Yeah, I guess just you
Okay.
maybe just use the arrows on the keyboard.
So I was interested in studying this because of my interest in parasitology and veterinary medicine. There is a lack of surveys studying the likelihood of parasitic crossover between wild and domestic species, especially in New York State. So I reviewed many studies in the Northeast on either wild or domestic species, but there were none studying both in the same study. It was also important for me to note that the other studies that I looked at were on a much larger scale, often over months or years with multiple researchers. This was important for me to keep in mind because I worked and collected samples alone and only worked for 5 weeks. So I collected samples from wild canines over the trails at Rice Creek, and this was an example of a week of surveying the trails. I changed the order of the trails each week, and so I would walk on the trails to survey them for wild samples in the morning, and at the end of the morning, I would collect samples from domestic canines from the dog waste receptacles at the field station and examine them in the lab in the afternoon for a total of 5 weeks. I collected 27 samples total, and I kept track of the date in which they were found and the trail they were found or the receptacle that they were found in, and whether I believe that they are wild or domestic samples based on field guides. I expected to find samples from wild canines that were either red foxes or coyotes. I followed the formalin ethyl acetate sediment concentration techniques outlined by Garcia in 2007. The first step was to mix 4 grams of stool with 10 milliliters of 5% formalin and mix it thoroughly. And then the mixture was strained through 2 layers of gauze into 15 milliliter centrifuge tubes. After that, a solution of 0.85% sodium chloride was added to fill the tube, and it was centrifuged for 10 minutes at 500 times gravity. Following the centrifugation, the supernatant fluid was decanted and the sediment was resuspended in 7 milliliters of 5% formalin and 4 to 5 milliliters of ethyl acetate. After that, I shook the tube for 30 seconds to mix the layers again and centrifuged one more time for 10 minutes at 500 times gravity. Following the final centrifugation, 4 layers would be visible in the tube, with the bottom layer being the sediment that contained the parasites. I decanted the supernatant fluid the final time to leave only a few drops of fluid on top of the sediment at the bottom of the tubes. That was then mixed with the sediment in the bottom of the tube and pipetted 1 drop from each sample onto a grid slide, along with Lugol's iodine solution to be the Lugol's iodine solution stain the samples to allow for a better view of the eggs and the larvae. I used grid slides to effectively count the intensity of the parasites, the larvae, and the eggs. And the pictures that I took were taken with a Canon camera attached to this light microscope. So the prevalence in parasitology is the number of infected hosts divided by the number of hosts studied in total. And the intensity is the number of parasites in an infected host. I used a range because of my limited data. This is an example of a diagnostic manual that I used to identify the parasite species. I compared my images to the images in the diagnostic manuals and compared the morphological characteristics as well as measurements. This table shows the results from my study. I included some phyla that are not included in the Helminths because I hadn't seen these 2 species included in any other study, and I thought that it would be important to include them. So the prevalence ranged from 1 in the domestic species. There were 2 different species found in the domestic samples with a 6.7% prevalence for each. And in the wild samples, there were 6 different species found with a prevalence ranging from 8.3% to 58.3%. So I looked further into the life cycles for each of the species found to further investigate the likelihood for crossover between wild and domestic canines. So for the hookworms, there was a prevalence of 7 out of 12 in the wild samples, so that's 58.3%. And the intensity range for eggs was 1 to 8, and the intensity for the larva was 1 to 28. And it's important to keep in mind that this was only 1 drop of the sediment from the centrifuge tubes. And the hookworms were not found in any of the domestic samples that were found. The hookworm's main mode of infection is accidental ingestion of eggs, but they also can penetrate the skin of bare feet of people or just the feet of animals. So this is an image I took. Eggs of all hookworm species are oval and contain morula, which is essentially an embryo. Several genera parasitize canines, but it's difficult to tell the difference between the genera just based on looking at photos. Here's a larva that I found. The second species that was found was whipworm. There was 2 found in the wild canine samples and 1 found in the domestic animal samples. Whipworms are also ingested normally as the main mode of infection, usually accidental ingestion of eggs. These do not penetrate through the skin as hookworms do, excuse me. And this is an image I took of the whipworm eggs. They're distinguishable by the polar plugs on either end of the symmetrical egg. And the third nematode species was the Toxicaricanus. There was a prevalence of 1 in each of the wild and domestic samples, with the prevalence in the wild samples being 6.7% and 8.3% in the domestic animal samples. The mode of infection for Toxicaricanus is similar to the other 2 species, but they also do not penetrate the skin, such as hookworm. And this was an image I took of the Toxicaricanus eggs. They're distinguishable by the rough and pitted outer shell with the deep pigmented embryo in the center. And then I also found some, what I believe to be 1 digenea species. The egg that I found does appear to have the same physical characteristics as the Fasciola hepatica egg. However, it's unlikely because the typical definitive host would be a ruminant rather than a canine. However, could be possible that a canine could have ingested raw beef liver in order to be infected with Fasciola hepatica. And this again is the egg. And I wanted to include some parasites that weren't in the Helminth category because they weren't in any of the literature that I had reviewed. So these 2 are in the phyla Apocomplexa. This is Isospora belli. It was found in 1 of the wild canine samples, and there were 4 eggs in that drop from the sample. And this is the life cycle. Canines may also just serve as the definitive host. It's here seen as a person. And this is Monocystis. It's actually a parasite of earthworms. It was found in 2 of the wild canine samples, presumably from the prey of the wild canines consuming the earthworms. And here's the life cycle in the earthworms with Monocystis. I also wanted to include some common artifacts that I found in the samples because they had not been mentioned in any of the literature that I reviewed. I had originally thought that some of these were parasite eggs, and it took a lot of research to figure out that they are most likely pollen. These 3 here on the left are most likely pollen, and then on the right there is pine pollen, and that was found in 11 of the 27 samples total. So I wanted to thank the Rice Creek Associates for funding my project and the Possibility Scholarship for providing me with housing when I was doing my research, and Dr. Andrew McElwain for assisting me during the duration of my research. Thank you. Thank you.
All right, thank you all very much. Thank you for coming. Thank you to the speakers. It's really good, really interesting talks.